Welcome to Click for IB Biology. As part of our genetics course, we're going to be looking at the phenomena of sex-linked genes. There's a blank PDF of this presentation down in the description. Download that, annotate with your own notes. There's a quiz, 10 questions with answers, and you can use that to check your understanding of the content given here. So let's start with biological sex before we move on to sex-linked genes. Biological sex is determined by the two chromosomes, the 23rd homologous pair, which are often referred to as the non-autosomal chromosomes. Okay. So here they are. Here we have, of course, in the biological females are designated XX. They have two X chromosomes as shown here. And then the male has one X and one Y. And by definition, the Y was inherited from father and the X was inherited from mother. Over here, we can't tell which one came from the mother or the father, but it came from one of them. When we look at a comparison of the size of the X chromosome, we see something which carries about 155 million bases and accounts for in excess of 1,000 genes, perhaps 1,500 genes. The Y chromosome, in contrast, is much smaller, known to carry about 55 million base pairs and to carry somewhere between 50 and 200 genes. The most famous of those genes is the SRY gene, which when expressed brings about the development of the anatomical male. Non-expression of the SRY gene leads to other conditions that students may well be very interested in exploring. Working on the basis of the two versions of biological sex, then we have the male here and the female here, and diagrammatically I'm showing the two chromosomes aligned with each other. And notice that where you have an XY chromosome, then this region here on the X chromosome has a homologous section on the Y chromosome, and therefore we could predict that for any genes in these locations here, they would have two alleles. However, for conditions genes which are encoding on this upper section, what we will call the non-homologous region, males will only have one gene, one allele for each gene. If we then compare that to the female condition, then with our two homologous X chromosomes, then every gene has a homologous partner, and therefore for every gene in females there are two alleles. If we apply this to a particular case, and our interest is not in the homologous region but actually in the non-homologous region, we find unusual ratios of conditions, of traits, of phenotypes in males and females. So we're going to look at an example, two examples, one of which is red-green color blindness and the other is of the condition known as hemophilia. The gene for red-green color blindness, the normal gene designated big B, is shown as X superscript big B. X lowercase little b is the mutated allele that causes red-green color blindness. Therefore, the potential female genotypes are X big B, X big B. This person would have normal vision. There is a heterozygote, X big B, X little b, and because B is dominant to little b, this person would have normal vision. And then the third phenotype is X little b, and this person would have the double recessive gene for color blindness, red-green color blindness, and this individual would be red-green colorblind. So that's an incidence of 2 out of 3 genotypes being normal, and 1/3 of the genotypes being colorblind. Now, if we compare that to males, because males only have one allele, remember that across here, the location of the red-green colorblind gene is in this region here, and with a normal allele, then this individual here would have normal vision, but this individual would have red-green colorblindness. So therefore, if we look at the ratio, we can see that the probability of having the red-green colorblindness in males in terms of the number of genotypes is about 50%. So red-green colorblindness then ends up being more common in males than it is in females. If we look at hemophilia as a condition, we're going to see the same phenomena shown, which is that the trait, the condition, in this case hemophilia, is going to be more common in males. This is often an early indicator of a sex-linked genetic condition. The alleles that we have for blood clotting is X big H. These people are able to, called normal, means that they can clot their blood when there are wounds. The X little h is an example of where the clotting factor is not being produced by the gene, and that would cause hemorrhaging, hemophilia, and bleeding. So let's take a look at the potential genotypes, which are shown here. So females with 2 X chromosomes and therefore 2 alleles per for the gene can be HH, that would be normal. Big H, little h, that's what we would call a carrier. So the person wouldn't actually have the condition of hemophilia, but they do have the allele, and they're carrying that potentially into future reproduction. And then we have X little h, and this person would be a hemophiliac and female. Now the interesting thing is that they are actually quite rare individuals, and we'll talk about that rarity in a moment. Our male genotypes are X big H, and that person would be normal, and X little h, and that person would be a hemophiliac. So as with the red-green colorblindness, here the chance of having the condition is 1 in 3 genotypes, whereas in here it's 1 in 2 of the genotypes, and so more common in males than it is in females. The rarity of hemophiliac females is because a male with, in order to become X little h, mother would probably be a carrier, but normal herself, and X little h would have to come from a father whose genotype would be X little h, Y. So as a daughter, she would inherit that one there. And this is until modern medicine allowed hemophiliacs to survive longer. Being a hemophiliac male would have been a selective disadvantage, and they probably would not have survived until reproductive age, and so having hemophiliac daughters would therefore have been unlikely. This table summarizes the genotypes and phenotypes of sex-linked conditions.